Imagine you wanted to map a huge, bustling city, but there's a catch. The entire city fits inside a single microscopic cell. Well, that's kind of the challenge we're tackling today. We're going to explore how scientists map that beautiful chaos inside all of us proteins. Just let that number sink in for a second. More than 10,000 different kinds of molecular machines, all working, all buzzing with activity in a space you can't even see. I mean, that's an entire universe packed into one cell. So where would you even start to map something like that? Well, you do it with a technique that creates maps that look a lot like this one. It almost looks like a star chart from a distant galaxy, right? But this is a protein map. Every single one of those tiny spots is a unique protein, sorted out from all the others and given its own special place on the map. Stick with me, and I promise by the end of this, you'll know exactly how this incredible star map is made. So here's our game plan. First, we'll talk about why this is such a big problem in the first place. Then we'll get into the main technique, a kind of molecular race. After that, we'll see what happens when you add another layer of sorting. Then we'll put it all together to see how these incredible maps are made. And finally, we'll look at why all this really matters. All right, let's dive right in with the sorting problem itself. Because to really get why the solution is so clever, you first have to understand just how massive this challenge really is. The best way to think about it is like this. Every cell is a microscopic city. And the complete set of all the proteins whizzing around in there, it's called the proteome. And it's not a quiet city. It's constantly building, repairing, communicating, the whole shebang. And if we can crack the code of this city, well, that's the key to understanding pretty much everything about health and disease. Okay, so how on earth do you sort out all the citizens in this protein city? Well, scientists came up with this really brilliant solution. They make them run a race, a molecular race. It's one of the most important techniques in all of biology, and it's called SDS page. The basic idea here is something called electrophoresis. You put your protein mixture into a slab of gel. It's kind of like Jell-O. And then you apply an electric current. That current is like the starting pistol. It forces the proteins to start moving through the gel. And how they move is what lets us tell them apart. But wait, there's a huge problem here. See, in nature, proteins are all over the place. They've got all these unique folded shapes, and they all have different electrical charges. Some are positive, some are negative. If you just tried to make them race like that, it would be total chaos. You wouldn't learn a thing. And this is where the genius part comes in. Scientists use a chemical detergent called SDS. Think of it as the ultimate equalizer. It does two incredibly clever things. First, it grabs every protein and forces it to unfold into a simple linear rod-like shape. And second, it coats every single one of them with a uniform negative charge. So just like that, you've gotten rid of shape and charge as variables. Now it's a fair race based on just one thing: size. And the process itself is actually pretty straightforward. You take your protein mix, you treat it with that SDS, load it into the top of the gel, flip on the electric current, and let them run. After the race is over, you add a stain, something like Kumasi blue, which sticks to the proteins so you can actually see where they all ended up. And what's really cool is that you can even tune the race course itself. You see, the gel is made from this stuff called acrylamide. And by tweaking the concentration, you can make the gels mesh tighter or looser. It's like adjusting the difficulty of the obstacle course. A loose 5% gel is great for separating huge proteins. A tight 15% gel is perfect for sorting out the really tiny ones. And this is what you get, these perfectly ordered bands. Each band isn't just one protein, it's a whole group of proteins that are all the exact same size. The smallest, quickest ones have zipped all the way to the bottom, while the big, heavy ones have barely moved from the starting line. The race is done, and our proteins are sorted, at least by size. But hold on a second. This brings up a pretty big question, right? What if you have two totally different proteins that just happen to be the same size? With this method, they'd get stuck in the same band, and we'd never be able to tell them apart. To fix that, we need to add a whole other dimension to our sorting process. And that's where another super clever technique comes into play. It's called isoelectric focusing, or IEF for short. Now, IEF totally ignores size. Instead, it sorts proteins based on something else entirely, their own unique natural electrical charge. How it works is, instead of a normal gel, you use a gel that has a pH gradient. It's acidic on one end and alkaline on the other. So you zap it with electricity, and each protein scoots along this gradient until it finds the exact spot, the exact pH, where its own charge becomes neutral. And right there, It just stops. It's found its isoelectric point. It's home. Now, remember that SDS we used earlier? It completely wrecks the protein's natural shape. So what if you actually need to see what the protein does? Study its function. For that, scientists turn to something called a native gel. It's a much gentler method that sorts the proteins without messing up their shape, so you can study them in their active, functional state. Okay, so now we've got these two really powerful tools in our toolbox. One sorts by size, the other sorts by charge. And this is where the real magic happens. What if you do them both, one after the other? Well, that's when you get a technique with some serious resolving power. It's a classic 1-2 punch. First, you do the charge separation. You take your protein mix and run it on a little IEF gel strip. Then, you take that entire strip, turn it 90 degrees, and lay it on top of one of our big SDS page gels. Then you run the second race, but this time you're separating everything by size. And boom, there it is. We're back to that incredible star map we saw at the beginning. Every single one of those spots is now an individual type of protein, pinpointed by its unique coordinates, its charge on one axis and its size on the other. The real breakthrough here is the resolution. This 2D approach can separate out thousands of individual proteins that would have just looked like a big ol' smear on a 1D gel. All right, so we've made this incredibly detailed, beautiful map. Now what? What's the point? How does this actually help us in the real world? Well, let's say you're not interested in the whole city. You just want to find one specific building or one specific protein. For that, you'd use a technique called Western blotting. Basically, after you've made your map, you use what's called an antibody. Think of it like a molecular homing missile that's programmed to find and stick to only one type of protein. It lights up just that one spot so you can find exactly what you're looking for. And this is where it gets really powerful. This is how these maps can literally save lives. In a clinical setting, doctors can take a sample from a patient, run one of these maps, and compare it to the map of a healthy person. Just look at this example from human blood serum. The lanes from patients with myeloma, a type of blood cancer, have this really distinct dark band that just isn't there in the healthy samples. That's not just a band on a gel, that's a fingerprint of the disease itself. And that's really the heart of it all. This isn't just about making cool-looking maps. This is the foundation of a whole field called proteomics. It's how we study the machinery of life on a massive scale. And it's absolutely crucial for understanding what goes wrong when we get sick. So we've journeyed from this invisible, chaotic city inside our cells to a detailed, readable map. And the better we get at making and comparing these maps, looking at the differences between a healthy person and a sick person, or between young and old, the more secrets about our own bodies we're going to uncover. We figured out how to read the language of proteins. So the big question is, what are we going to find out next?